Hello again, Bio 220 students, and welcome to this installment of Online Microbiology. This chapter will cover microbial growth. Now, when it comes to microbial growth, there are various things that can affect how effectively or how poorly microbes will actually grow. And some of them can be physical, such as temperature, the pH, whether it's too acidic or basic, as well as osmotic pressure, how much dissolved solutes are inside versus outside the cell. There are also chemical necessities needed for cells to grow properly, including a variety of elements that we'll get into. Now, as far as physical requirements, temperature, there is basically three situations. There is a minimal temperature needed for a certain microbe to actually grow. There is a maximum temperature beyond which it will die. And then there's an optimal temperature at which the growth rate is at its maximum. Now, as far as three different types of microbes, there are psychrophiles, which love colder temperatures, mesophiles, which like moderate temperatures, such as around our body temperatures, and then there are thermophiles, which are heat-loving. This graph just shows how certain bacteria or certain types of bacteria have preferential temperatures in which they can actually grow, and otherwise they will, and outside these regions, growth is not typically possible. Getting into psychotrophs, well, they typically grow between 0 degrees Celsius or the freezing point of water to around 20 and 30 degrees, so around room temperature. And they typically will result in food spoiling. This is just another way of representing how many different bacteria have certain optimal temperatures. And below optimal temperatures, growth will typically slow down and stop, and extreme heat will usually kill off most microbes. Now, as far as temperature goes, there are thermophiles, which generally have a preferential growth temperature around 50 to 60 degrees, and they'll be found in sources of hot springs as well as decomposing organic matter. There are hypothermophiles, which greatly appreciate temperatures above 80 degrees Celsius, where around 100 degrees Celsius is the boiling point of water. Now, pH, this deals with essentially how acidic or how basic a solution or environment is. Most bacteria will typically grow around pH 6.5 to 7.5, so around biological pH of our bodies. Molds and yeast will typically grow at a slightly more acidic environment between pH 5 and 6, but there are acidophiles which grow in very acidic environments, so very, very low pH. And with osmotic pressure, hypertonic environments are where the amount of dissolved material is higher in concentration outside the cell than inside the cell, and these situations can cause plasmolysis due to high osmotic pressure. When in the case of extreme or oblique halophiles, they actually do require a high osmotic pressure or an environment with a lot of salts. And for the case of facultative halophiles, they will tolerate high osmotic pressure, so a high difference in dissolved solute concentration. And plasmolysis, that's just simply where the liquid portion of a cell is drawn out of the cell, typically causing the cell to shrivel on the inside. Now, as far as chemicals or elements that are standard for life Carbon will be the most common one. It is the structural background or the major component of most or all organic molecules used for life. There are chemoheterotrophs that actually use organic molecules as energy, where in the case of autotrophs, they use carbon dioxide instead. Nitrogen is another common necessary element. It is a primary component found in proteins as well as DNA and adenosine triphosphate. Most bacteria will decompose protein material to obtain their nitrogen. And there are some bacteria that instead use ammonia or ammonium ions and nitrate ions as their source of organic nitrogen. And there are a few bacteria that actually can use molecular nitrogen, or N2, in their nitrogen fixation, where fixation means the process of incorporating that element into necessary biomolecules. Another important element would be sulfur, which is also used in amino acids, but also thiamine and biotin. It's used, or most bacteria that will decompose protein as a source of sulfur as well, in addition to nitrogen. And some bacteria will instead use sulfates and hydrogen sulfide. And phosphorus, it's used readily in DNA, RNA, and ATP. It's found in the phospholipid membranes of cells, and phosphate is a typical source of phosphorus for many microbes. There are trace elements, which are inorganic elements that are required in small, minute amounts. Usually they are involved in enzyme cofactors, and these can include a variety including iron, copper, molybdenum, as well as zinc. In the case of oxygen, oxygen can affect a variety of microbes in different ways, sometimes in beneficial ways and sometimes in hazardous ways. As far as the different types of microbes in regards to how they use oxygen, there are obligate aerobes, which need oxygen in their environment in order to grow. There are facultative anaerobes, which will grow via fermentation or anaerobic respiration when oxygen is not readily available. There are obligate anaerobes, which are unable to use oxygen and are harmed by its presence. There are aerotolerant anaerobes that will tolerate or live in oxygen environments, but they cannot actually use oxygen itself. And then there are microaerophiles, which require oxygen concentrations, but at concentrations lower than in our present air. And this is just a chart detailing the variety of microbes that were discussed. Now, oxygen can actually be hazardous because it has a tendency to form different radical or unstable compounds. One example is a singlet oxygen, which is a highly energetic and reactive version of molecular oxygen. There are superoxide radicals, as well as peroxide ions and hydroxyl radicals. Essentially, these are molecules of oxygen that are very unstable and can do a lot of damage to living cells. As far as organic growth factors, there are organic compounds obtained from the environment, and these can be vitamins, amino acids, as well as purines and pyrimidines used in genetic material. Now getting into biofilms. These involve communities of microbes, and they can be slime or hydrogels that are formed by those communities of microbes, and they will help the bacteria adhere to surfaces. And they also allow a sort of quorum sensing between communities of microbes, where essentially certain molecules will be produced in higher amounts as the density of that bacteria population increases. It allows for a sharing of nutrients, and it also protects bacteria from harmful environmental factors. Now, as far as biofilms are found, they can be found in the digestive system as well as in sewage treatment plants. And as biofilms are formed by microbes, they can clog pipes. And they are 1,000 times more resistant to microbicides, or things that would kill microbes on their own. And they are typically involved in around 70% of all infections, involving catheters, heart valves, and other important devices and parts of the body. Getting into actual media, which is essentially a substance used for actually preparing a culture or a lab sample of bacterial colony, or microbial colony. There is the culture medium, which is essentially the nutrients prepared for that specific microbe, needed for that specific microbe to grow optimally. There is also a situation called a sterile environment, which means no other microbes are currently in that media. And then inoculum is when you introduce the desired microbe into that nutrient medium. And then the culture is the term for when microbes are growing or on a culture medium. Essentially, you are growing a colony of that microbe you desire. Another substance involved in media preparation is agar. It is a complex polysaccharide, and it's used as a solidifying agent for culture medium in petri plates, slants, and deeps to essentially stabilize it and keep it in a nice solid state. Generally, microbes will not use it in metabolism, and it will liquefy at the boiling point of water, and it will solidify at around 40 degrees Celsius. And 40 degrees Celsius in Fahrenheit is essentially 100 degrees or 104 degrees Fahrenheit. So at room temperature, agar is reliably in a solid state. Now, there are chemically defined media which have an exact chemical composition that is well known. Fastidious organisms are those that require many growth factors provided in chemically defined media. So essentially, this is where a certain recipe is used for a media to grow a specific type of microbe. There are complex media where extracts and digests of yeast, meat, or plants and chemical composition will vary from batch to batch and involves two terms, nutrient broth and nutrient agar. This is just one example of, say, a typical media for growing, or a typical recipe for a culture media to grow a culture of E. coli. Again, the media will vary from time to time, depending on the microbe. Now, for anaerobic growth media, where oxygen should be avoided, there is what's called a reducing media. It's used for the cultivation of those anaerobic bacteria, and it will contain chemicals like sodium thioglycolate that combine with oxygen to help remove it or use it up. And the media will be heated to also drive off dissolved oxygen. This is an example of a sealed environment to help keep out oxygen while growing anaerobic sensitive bacteria. And this is another situation, another form of equipment, which is a gas chamber that essentially keeps out the undesired atmosphere, whether it keeps out oxygen or fills a atmosphere with pure nitrogen. It can range from what is in there, but essentially it's a way of working with microbes that are sensitive to the compounds or molecules common in our atmosphere. There are special techniques for preparing culture. In the case of capnophiles, they require high concentrations of carbon dioxide, which in reality is about less than 0.5% of our natural atmosphere. So in those situations, a carbon dioxide packet is needed to add carbon dioxide, as well as utilization of a candle jar to keep the environment sealed. There are hazards to keep aware of, such as levels of biosafety, where essentially they range from 1 to 4, where the first is there are no special precautions, and basic teaching labs will use those levels of microbes. Biosafety level 2 would require the use of a lab coat, as well as gloves and eye protection to maintain safety. On the third level, biosafety cabinets, or what was shown before, are used to prevent airborne transmission of certain dangerous microbes. In the case of biosafety level 4, a sealed negative pressure environment, or a hot zone, is involved. It will exhaust air that is filtered twice to maintain, or through HEPA filters to make sure that nothing escapes quarantine. This is an example of working with a microbe that is significantly more hazardous and requires level 4 safety techniques. Now, there is a type of media called selective media. It suppresses unwanted microbes, but encourages the desired microbe. And it will contain inhibitors to suppress growth for those that are not desired. There is also differential media, which is a media that is specialized to allow for the growth of various different colonies. And it allows distinguishing those different colonies of different microbes while on the same plate. Some media will have both selective and differential characteristics to both keep out unwanted microbes that might be common in daily life, but allow for a select few to grow and be compared. Bacteria colonies will typically show up in some grouping. What is shown here is essentially a differential media where 1/3 is used as a control to show no colonies of microbes being grown. Therefore, it is not inoculated, where the bottom left is filled or was inoculated with Staphylococcus epidermidis, and the bottom right was inoculated with Staphylococcus aureus. And this is just a way of observing different types of microbes on the same media. Enrichment culture. It is something that encourages the growth of the desired microbe by increasing very small numbers of a desired organism to detectable levels, and usually it will involve a liquid. And this is just another way of grouping the different types of culture media. Getting into actual colonies. A pure culture contains, it's desirable to only contain one species or one strain of microbe. Now, a colony is a population of cells arising from a single cell or spore, or from a group of attached cells. A colony is often called a colony-forming unit, or CFU. And the streak plate method is used to isolate pure cultures. For example, where essentially bacteria or microbes are added in a streaking fashion to eventually see where pure colonies are forming as individual spots. Preserving bacteria cultures so that they can be maintained for a later date can involve deep freezing, which is involved using significantly cold temperatures well below the freezing point of water, typically -50 to -90 degrees Celsius. There is also lyophilization, or freeze drying, where essentially the bacteria culture are frozen and then put into a vacuum, which quickly evaporates or sublimates any amount of water that was frozen. And by sublimates, it evaporates it off into the gas state, essentially. When it comes to bacterial division, this is where there is an increase in number of cells, but not cell size. One example is binary fission, and through the process of budding. There is also candidiospores, such as actinomycetes, and then fragmentation of filaments. In the case of binary fission, essentially the DNA has the cell elongates and the DNA is replicated. The cell wall and plasma membrane begin to pinch off, and this is where we start to see two new cells starting to form. The cross wall forms, or essentially the divider, forms and completely separates off the two strands of DNA. And then eventually the cells separate to become two distinct cells. When it comes to generation time, this is the time required for a cell to divide, and these can range depending on the microbe from as easily on the order of minutes to a day. Binary fission doubles the number of cells during each generation. Therefore, to get a total number of new cells or a total population after a certain amount of time, that would just be 2 raised to the power of the number of generations that have been allowed to pass. Growth curves are used to represent this growth in a logarithmic fashion. Essentially, a log function is a way of taking some exponential growth data and represent it more in a linear fashion as far as displaying it on a graph. Because anything that could be represented in some linear function is easier to plot out and interpret. Now, phases of growth. There are going to be 4 different phases. Essentially a lag phase, which is the intense activity of a cell preparing for growth, but there is no growth in the population yet. There is the log phase, or the logarithmic or exponential growth or increase in that population of microbe. Then there's the stationary phase, which is a period of equilibrium where microbial death is balanced by the growth of new cells. And then there's a death phase, where the population trends to decreasing at a logarithmic rate. For direct measurement of microbial growth, that's a way of counting the amount of microbial cells. It involves plant count, filtration, most probable number estimate method, MPN, and the direct microscope count. For plate counts, colonies can be counted on plates that have around 30 to 300 colonies. To ensure the right number of colonies, the original inoculum must be diluted via serial dilution. And then counts are performed on bacteria mixed into a dish with agar, and then a pour plate method, or a spread on the surface of the plate, which is the spread plate method. Serial dilution, that is just simply taking one original solution of a bacteria, taking a little of that first solution, putting into a new tube, and diluting the volume, then putting a little bit of that second solution into a third vial, and then diluting the volume, and slowly decreasing the concentration of that microbe. Filtration. Filtration, it's a solution is passed through a filter that collects the bacteria as the liquid component continues through. The filter is transferred to a petri dish and allows for growth of colonies on the surface. The most probable number method, or MPN, involves multiple test tubes, multiple tube tests, count positive tubes, and compare with a statistical table. There is the direct microscopic count. A volume of bacterial suspension is placed on a slide. The average number of bacteria per viewing field is calculated, and what is used is a special Petrov-Hauser cell counter, which essentially is the number of bacteria per milliliter is equal to the number of cells that were counted, divided by the volume of area counted. And then estimating bacterial numbers by indirect methods. One could be turbidity, which is a measurement of the cloudiness with a spectrophotometer, an instrument that's used to shine light or beam light through a sample and then determine how much of the light is able to pass through that sample to a detector on the other side. There is metabolic activity, which is the amount of metabolic products or substances that microbe will produce, and see how proportional it is to the number of bacteria. There's also dry weight, which is when the bacteria are filtered, dried, and weighed, which is good for filamentous organisms. This is an example of using a spectrophotometer, where again light is shown through a sample, and a certain amount of that light will eventually make it through the sample, whereas some or most will not be able to pass through and are blocked by the bacteria in that suspension. It involves using a blank first to show how much light a detector can expect if there is no microbes in there, and then how much light will pass through in a second sample that does have microbes. That way the difference can be determined. And that concludes our lesson into Chapter 6, which is our lesson in microbial growth. See you all in the very next lesson video.